So the rules for RNA synthesis are very much like those for DNA synthesis. And if you've studied cellular DNA synthesis, which I assume you have in a biology course, then all of these should be familiar to you. RNA synthesis begins and terminates at very specific sites on the template. It can be at the end or it can be in the middle. It can initiate de novo. That means without a primer. And cellular DNA-dependent RNA polymerase, the enzyme that makes messenger RNAs in the cell, that initiates de novo. It doesn't need a primer. But some viral polymerases need one. And the ones that do, you couldn't predict. So you just have to know that some of them do. In addition to the RNA-dependent RNA polymerase, other proteins are needed for polymerization, and they can be viral in cell, and we'll talk about some of those. The synthesis of RNA, of course, is directed by a template, with one exception, which we'll see at the end of this lecture. Template directed, which means you have a RNA as a template, and then the RNA polymerase reads the template and puts the complementary bases. And of course, it synthesizes RNA in a 5' to 3' direction, just like DNA. So the template is read in a 3' to 5' direction. And as I said, we have some non-templated RNA synthesis. It's not the majority of occurrences. It's an exception, but it's quite interesting, and we'll talk about that later. So here are an example of some kinds of initiation, just to give you a feel for how this works. So de novo initiation, in other words, you don't need a primer. The polymerase can recognize the template and just start putting complementary bases down. So you can see here, here's the template, this green molecule. This is a 3' end. That's where it starts, the polymerase starts to read. And then the polymerase would just put the first complementary base down. It's beginning right at the 3' end. So that's de novo initiation. You don't need a primer. And as I said, some polymerases can do that, others cannot. Here's another example of priming de novo. In this case, instead of a nucleoside or an NTP, it's a capped NTP, but it's the same principle. And in this case, there's internal priming, some synthesis, and then the primer slides back to make a perfect match. The point here is that you don't need a primer. These are just two examples. Then we have primer-dependent initiation. And the viruses we will be talking about have two different kinds of primers. They can have a protein-linked primer. So it's a short piece of complementary RNA. That's what a primer means, right? It can be very short. In fact, in the case of the protein-linked primer, it's 2 bases with a protein linked onto it. We'll talk about this today. Or the primer can be capped. And we'll again talk about this. And by capped, I mean it has the unusual 5' base linked by a 5' to 5' linkage to the RNA. This is typical of cellular mRNAs. And some viruses use those with about 10 or 13 nucleotides beyond as a primer. Right, so de novo or primer-dependent initiation by these polymerases. So you're one or the other. They don't do both. So it's either primer-dependent or not. And again, this is the mechanism of actually DNA synthesis. You can see this is DNA here because we have T's being put down, but it's the same mechanism for RNA synthesis. And you may have encountered this in a course here. This is the so-called 2-metal catalysis model of nucleic acid synthesis. So here is the general situation we're looking at here. We're looking at a template in the black line. There's a primer here in red. And synthesis occurs by adding stepwise the triphosphates that match up with the template. And here we have a template on the right from 3' to 5', and here are the individual sugars, the ribose sugars, and the bases, G, T, A, T. And you can see the polymerase is adding bases, synthesizing them in a 5' to 3' direction, C, A, and T. And the important point here is that at each polymerization step, the catalysis is assisted by 2 magnesium ions that are coordinated by 2 aspartate residues. Here you can see aspartate A and aspartate C. And you can see these are coordinating the magnesiums, and these assist in the catalysis so that the nucleophilic attacks can occur and get rid of the 2 terminal phosphates and link up the new base in here. So this new base has just been covalently linked, and we're going to release, of course, a diphosphate or a pyrophosphate at that step. So the two-metal catalysis model serves for both DNA and RNA, and we're going to see where this happens in the polymerase. Now, every polymerase that we know of, cellular or viral, is related in some way. And this is a diagram that lines up the sequences of 4 different classes of polymerase. So we have RNA-dependent RNA polymerase, which is our viral polymerases that we'll be talking about. We have RNA-dependent DNA polymerase, that's reverse transcriptase. And we have DNA-dependent DNA polymerases. And then DNA-dependent RNA polymerases. These are the enzymes that make mRNA. So these are all related. The green areas show the areas of the greatest homology. And these areas that are shaded A, B, C, D, and E, these correspond to structural motifs on the enzyme. And I'm going to show you that in a moment. The structures of members of all of these 4 categories of polymerase have been solved. And you can see why they're related. So the fact that they're all related probably means that they evolved from a common precursor. Right? Many years ago, there was probably one polymerase. Maybe it was an RNA polymerase in the RNA world, and then all the others evolved from that. A couple of things to point out. As I said earlier, you can identify an RNA-dependent RNA polymerase by this GDD signature, GlyAsp-Asp, in one of the motifs, in the C motif. The asp-asp, those are the 2 asps that coordinate the magnesium ions that are necessary for catalysis that I just showed you. So in all plus-strand RNA polymerases, you find this GlyAsp-Asp. So if you identify a new virus from a sick person, for example, and you sequenced it and you find a GlyAsp-Asp, that would be a good sign that it's a plus-strand RNA virus. The negative strand RNA viruses have slightly different motifs. They have an asp-asp. In the negative strand RNA polymerases of segmented RNA viruses. Reverse transcriptase also has an asp-asp. And again, those 2 residues crucial for coordinating the magnesium. And non-segmented negative strand polymerases like measles virus, VSV, they have a GlyAsp-Acin. So the asp-acin coordinates the metals. Just to show you that you can not only identify these polymerases by these sequences, but they have an important catalytic role in coordinating those magnesium ions. Now here is the 3-dimensional structure of the poliovirus RNA-dependent RNA polymerase. So this is a plus-strand RNA polymerase. It was the first RNA-dependent RNA polymerase structure to be solved, and when it was solved, it was the last of the 4 to be solved, and it fit right in. It aligned up perfectly with these motifs, and it was clear that all 4 of them were related. Now you can see here I've labeled the different parts of this polymerase, A, B, C, D, E, and F, and those correspond to these shaded motifs here, and you can see that they are very specific parts of the polymerase. For example, the yellow, C, which is right here, that's the part that has the GlyAsp-Asp, and here are those 2 aspartate residues right there. And those are the ones that coordinate the magnesium. So that tells you right away, this is the active site of the polymerase, that yellow strand right there. And all these other motifs, the A, the D, and so forth, are conserved as well. These polymerases look like a right hand with fingers and thumb domains, and the palm in the middle is the active part of the molecule. So all the polymerases have the same similar motif, a right hand. Now, a couple of things I want to point out about this structure which are really interesting. This is an RNA-dependent RNA polymerase, so it makes RNA from RNA. If you give it DNTPs, deoxynucleoside triphosphates, it will not use them. It only wants to use NTPs, the ribo-NTPs, not DNTPs. And the reason is shown by the structure. So this is a part of the active site we're zoomed in. Here's the 2 aspartate residues, again, that coordinate the magnesium. So this is the active site. And here is a triphosphate, an NTP, not a DNTP, an NTP that's about to be added to the growing chain. So this polymerase is copying a template, and this is the next NTP sitting right near the active site, ready to be added. And the way this works, by the way, is it tries all the 4 different ones, and the one that matches up is most energetically favored, and then it gets added. It has been so quickly that it can do it that way. So it doesn't know ahead of time, of course, what base should go in here. But this NTP, you can see, forms various types of interactions, a lot of hydrogen bonding, for example, with neighboring residues. These are amino acid residues. And this one is very important here. This interaction with this asp at 238. If this were a DNTP, there would not be a hydroxyl here. It would be a hydrogen, and it couldn't form this interaction here. So that's why this enzyme only uses NTPs, because of this amino acid here. You can actually mutate that amino acid and allow it to use DNTPs. So here's a space-filling view of the enzyme with product and template. So this is in the process of polymerizing. We've actually cut away a bit here so you can see in. Here's the active site, and the purple here, those are the 2 aspartate residues that have been shaded purple, and this is one of the magnesium ions in there. Okay, so that's the catalytic site. That's where the NTPs are added to the growing chain. So the template is blue. You can see it comes in through the bottom. We're actually looking down on the top of the enzyme here. So this is the front face. This is the top. The template comes in here, goes through the active site, and then comes out the top. And here the product is being made. That's in gold. And the next triphosphate will be added here. So from solving the structure of these enzymes, we can really understand a lot. And I've just told you a little bit about some of the properties, but we can learn a great deal about how this catalysis occurs.